Top Bed Talk.
Monty Mython here. As we prepare to move into the next phase of the COVID crisis, we're going to be widening our focus here on Top Bed Talk. We will continue to deliver COVID-specific programs, but we all have a huge additional responsibility as we try to reboot normal services. So although this piece is not directly related to COVID-19, we believe it's information that is crucial to the bigger task of rebuilding the healthcare system as we learn to adapt and live with this new virus. Thank you to our sponsors and to you, our listeners, for helping us to share this important information.
This piece is presented by John Whittle, Assistant Professor at Duke University Medical Centre.
So, lots of the talk we've had this weekend has been about, I've heard the word sarcopenia, we've talked about delirium, we've heard inflammation, we've heard a whole bunch of reasons why people might do badly after surgery. And I apologise to people who came to our CPET workshop yesterday. You may recognise some of the slides from this. But what might a low aerobic exercise capacity signify? And I'm going to try and encourage us to move from organs, heart, lung, single organ complications, to maybe thinking about why the cell results or cellular dysfunction might result in multi-system pathology. So I'll give you the summary of the talk first, so if you all fall asleep, you got the answers. Low aerobic exercise capacity is associated with impaired perioperative outcome. That is confirmed. Post-operative complications are multi-system and distant from the operative site. That means when you make a hole in the gut, you get pneumonias and AKIs and other such things. Reduced cardiac output. We love it this side of the pond, don't we? The heart and cardiac complications. It's really only part of the story. What about low aerobic exercise capacity? Well, it's associated with inflammation. It's associated with mitochondrial dysfunction. It's associated with autonomic dysfunction and probably inter-organ crosstalk. And associated with altered substrate utilization. So we've also heard prehab talked about a lot today. It's effective. How is it effective? Why is it effective? How can we target it in the time that we have before surgery? So I'm going to sort of bring up this concept of the difference between exercise and training, right? So just doing more movement versus training for your marathon, which is the major surgery that you're going to undergo. So who develops post-operative complications? Well, these are sort of the ones that we, again, have heard a lot this weekend. So sarcopenia, malnourished people, frail people, what do these things mean? They're debatable, people with metabolic syndrome, diabetes, they get lots of complications, don't they? Hypertensives get complications. We kind of nebulously talk about inflammation and immune status pre-surgery or nod sagely. I do the same. Physiological age, I write physiological age and not chronological because I think there's a difference. Things that tie all these things together are a low peak VO2 or poor aerobic exercise capacity. So, multiple systems remote from the operative site, multi-system drivers influenced by disrupted physiological control. And when we sit there in the pre-operative area or in the clinic or so, and we look at these patients, we're terrible at, and it's proven, we're terrible at going, you're going to get all the complications. It's not something we can just do by looking at patients. So, we need finer tests to really decide who we really need to focus these efforts on. So, I don't really want to spoil the slide. Have you already revealed the results of the Slido? Oh, I'm sorry, guys. Don't look if you want to participate in the Slido poll. Where does morbidity occur? I.e., I say it's distant from the operative site. Well, it really is. So kidney dysfunction, infection, pulmonary complications, cardiac's quite low down there, gastrointestinal. This is for a whole bunch of surgeries, the orthopedic, general, urology, vascular, but not necessarily all the time where we make the hole. And what about single organ complications? We love looking at, why do we get a heart complication? We get a lung complication. It's an integrated organism, so we should treat the human being as such. Now, this is a slide from my colleague Tim Miller and Scott. This is the central premise behind ERAS. And again, I don't want to labour the point, but I'm going to. surgery is quite complex in terms of what it does to the human individual. So there's a stress response. We all know that. We try and mitigate that. ERAS tries to mitigate that. You get, SIRS response. Your pre-existing lymphocyte and cellular function will influence how you do. Your mitochondrial function will influence how you do. Pain feeds into all this mix. Protein catabolism, and if you start off poor, what happens to you afterwards? Important. Immobility, let's tie that in. Insulin resistance, and cardiac dysfunction. So solid surgery is a multi-system challenge. So really, what we're looking at is a marathon. So when your patient turns up for their main event, the marathon, the hemicolectomy or the AP resection or whatever it is, it's their marathon. So we're looking at treating our patients like athletes. How can we train them? So the current paradigm is this. So why does aerobic exercise capacity relate to poor outcome? Well, it's because we've got to increase our oxygen delivery to meet the oxygen requirements associated with the increased metabolic work required to survive, i.e., that's why. And certainly there's some truth in it. After surgery, there's an increase in oxygen consumption, and the stress response or repair after surgery is a stressful event and requires energy. But it's not the whole story. And this is kind of why at the moment we all sit there in the CPET world, the cardiopulmonary exercise testing world, and say aerobic exercise capacity equals complications, because you have to. If you can't increase your oxygen uptake when you exercise, then you definitely can't do it after someone makes a hole in you. True, but maybe the other way around. Not the whole story. So, what about the patients we're seeing? So we're seeing an increasingly elderly population. We're seeing increasingly comorbid people. And we all know that one 75-year-old isn't the same as the other 75-year-old. And sometimes we can't quite put a finger on it. We've looked at lymphocyte subsets, neutrophil lymphocyte ratio, aerobic exercise capacity. But really, these patients who are getting the complications are possibly the ones who have been subject to this inflammaging paradigm, accelerated aging, they have these age-related diseases. What are the biological hallmarks of aging? This all ties into what Mars has been talking about as well. You've got to take your choice. So I've decided to go with mitochondrial dysfunction as being one of our key issues in surgery. So going back to it, why is this guy ahead of us? Okay, there may be another fat 50 diabetic who comes to your clinic who gets the complications. This guy is probably going to do all right, okay? So, at the moment, certainly the other side of the pond, and in Australia, cardiopulmonary exercise testing is used quite a lot. It's based on this recognition that integrated physiology is more important. Our patient isn't just a heart or a pair of lungs when we put them on the table. So why would you only measure an individual organ system and then try and predict outcomes afterwards? But as I said, we're still quite fixated on peak VO2, and those of you who are familiar with the MET study that came out relatively recently, peak VO2 seemed, was the big readout from CPET. And I'm going to sort of say it's probably true, but we'll discuss a bit why. So where's the limitation? So again, we all learn at medical school or physiology school or wherever you learned your physiology, if you paid attention those days, that when you exercise, the heart is the limiting factor. And so that's why we're all so interested in pump function. So I am your heart. What do I do? It pumps blood. Okay. But we've already discussed in this meeting a little bit, this MINS paradigm, and it doesn't seem to be related to thrombosis, it doesn't seem to be related to perfusion. So I would argue that actually that's because it's reflecting the cellular milieu throughout the body, not just, endothelium or this, that, or the other. And certainly that seems to be true. So, for example, in a publication from the group I was formerly a part of last year, looking at the vision cohort, the original MINS paper, myocardial injury after non-cardiac surgery, 40% of patients with established inflammation developed MINS, and if you had the MINS, then you were more likely to die. So, you're already having a look at this sort of concept that your established cellular phenotype not only will predict whether you get an troponitis, that sounds a bit inflammatory, doesn't it? But whether you survive it. So where's the limitation? Mitochondria. This is what a mitochondrion looks like, by the way, not those pictures you see with the double, Right. So what about this paradigm about aerobic exercise capacity being limited by the heart? It's kind of true. So if you're an elite athlete, yeah, cool. There's no correlation between your whole body VO2 max and your mitochondrial VO2 max, i.e. perfusion is the limiting factor, your heart's the limiting factor. If you're not, like most of our patients, an elite athlete, then actually there's a very strong correlation between your mitochondrial oxidative capacity and your whole body VO2 max. So the heart is the slave of the cell and not the other way around. So don't think of it, the heart pushing oxygen, the cells are demanding it. And then what can they do with it is the key, right? So how are we using it? I'm conscious we're short of time. How are we using this concept? Well, we're looking at using circulating cells, so circulating immune cells, lymphocytes, monocytes, platelets. These cells go around the body. They get exposed to the physiological milieu in the heart, in the kidney, and wherever, because that's their nature. And they reflect the mitochondrial function in the organs that they pass through. There's cardiomyocyte, skeletal muscle, the neuron. And what about the substrates they're using? So when you look at it, you don't just look at the oxidative capacity, but what are they able to use? So we know that if you're deconditioned, when we exercise you don't use fat as a fuel, or you start off, so the lower graph there shows that, start off using a little bit, and as soon as we stress you switch to sugar. And we can train that. The more training you do, the more likely you are to use fat at lower metabolic rates, lower levels of intensity. And if we stress you, you're more likely to move back to using sugar. So what about that guy, that fat 50 diabetic guy? I'd argue his muscles look a bit different from the muscles that we see here. So on the left-hand side of the screen, we have a normal muscle. These two patients would have the same BMI, okay? The one on the right is an obese patient with a low muscle volume and a high fat infiltration. And again, in our lab, we're using ultrasound or muscle ultrasound to try and look at muscle quality. So this is muscle sound is the name of the technology. And it gives us intramuscular adipose tissue, muscle volume, an idea about glycogen content and muscle architecture. And what do we know about muscle fat? Well, it's dynamic. So if you're septic, your muscle quality changes. So on the far right, we have a young 20-something-year-old guy who unfortunately developed some quite severe sepsis. You can see his muscle volume. If you look at him, he's not sarcopenic, but the muscle quality is poor. It's full of fat, and it's full of increased tissue interfaces which reflect connective tissue, so that orange is fat. This obese patient with sepsis, certainly compared with obese control, has more fat, but is actually possibly not as badly off, you could argue. Same over time with neurotrauma. This is a chap whose head has been hit, and over time, his muscle has atrophied, and the content of fat has changed. Last minute, the fat in the muscle is complex, so athletes do have intramuscular fat. So if you look at a Tour de France cyclist, you will, on any given time, you'll see a high fat content to a low fat content to high fat content. So the ability to use the fat and cycle the fat is another important thing. So this is a dynamic thing. Obese people, or type 2 diabetics, have high fat content but are unable to recycle it. So one of the things is, I'd like to say is that when we're talking about sarcopenia, we're not just talking about a low muscle volume. We need to be looking at the muscle quality and muscle function. And when I talk about muscle function, I'm talking about aerobic exercise capacity as a reflection of aerobic exercise. And when we're training patients, and we're training them, not just exercising them, not just telling them to walk, what we're doing is we're altering the muscle. The muscle sends out these myokines, they alter immune signaling. We're altering the fat reuse, just like in our athletes, and that also alters the immune inflammatory milieu. We're therefore changing the inflammatory milieu, and actually that's possibly why in some of our colleagues in Southampton, Professor Mike Grocott studies, the cancer outcomes after prehabilitation are improved. So I already showed you the summary at the start because I knew I'd run out of time. So I'd just like to thank my colleague Euron Mullinger, who's sitting at the front here, who's brought a lot of this technology with us, and David McLeod, who's the director of the lab that we're doing a lot of this work in. Thank you.
Top Med Talk.
Nick Majerison here. Have you got yourself onto EBPOM.org yet? If not, you might not be aware, EBPOM Chicago. Tickets are free for a limited period only. Go now to EBPOM.org. Evidence-based perioperative medicine. EBPOM.org.